Today we're going to have an overview of the HumanCellAtlas project, which would be done immediately after my remarks. We would have some scientific presentations under the session highlights of single-cell research and discoveries in Africa. Then we'll have experimental design issues and discoveries, which are also going to be scientific presentations. And we're going to have ethical issues and discoveries, technical issues and discoveries, and then we'll close for the day, hopefully close on time. Then for tomorrow, we're going to have a session on HCA as a reference for disease research. We're going to have an interactive session on opportunities for collaboration, and then also future directions for African single-cell research. And this will be through a panel discussion. After that, we'll have another panel, Ask Me Anything panel. And then we'll have the next steps town hall, where we will talk about where we go from here and what we can take away from this, this symposium. Then we have the meeting summary and closing remarks. So that's for tomorrow. Just some notes about using the platform. So when we finish one session and we need to move on to the next session, there's a break in between, but then you have to close the window and then go and click on the next session from the program, and that will take you to the next session. So basically, it's not going to be a continuous session. After each session, you have to close the window and go back to the program and click on the broadcast link and go to the next session. We're going to keep it very lively on social media. So if you look, you'll see that there's a social media link or social wall link where you can engage and also see what others are saying. So you can follow the conversation using the hashtag HCA 2022 Africa. So please, let's keep it lively. There's also the option to interact with colleagues. So you can go into your profile and add your picture, and then you can be able to see others as well. Don't forget to save after you've made any edits, so that the edits are saved if it's changing your picture or editing your profile. There's also a resources page where you can find very important information about HCA, including the data release policy, the white paper, and then some links to training material, publications, videos, and more. You can also get a link to HCA's commitment to humanity. So these are all resources that you can enjoy whilst the workshop or the symposium is ongoing. There's a help desk link to that you can use. Should you have any issues or challenges, you can go to the help desk and ask questions, or you can go to the frequently asked questions, where you can have answers to some of the issues that are commonly encountered. So you can use either the help desk or the FAQ. And then you have the chat and then the question and answer options also. So to chat to all meeting attendees, use the room chat option. But if you wanted to ask questions of the presenters, or if you want me to see your questions that I can then pass on to the presenters, then use the Q&A option. So please, let's use that. Once the presentation starts, you can start putting your questions there, and I'll read them out as we go on. You can also raise your hand if you wanted to speak. We have that option. So if you raise your hand, the moderators will bring you into the room and you can speak and we would hear you. So if you don't want to use the chat and you want to actually speak, please use the raise your hand button, and then you will be let into the room. So a little plug for the app we're using, Disable. So if you want to try it after this, you can download it and you can download it from the App Store or Google Play, and it's called Disable. So, all right. So we want to appreciate the Chan Zuckerberg Initiative for part of the support for this symposium. They have provided funding for this, and we're grateful to the Chan Zuckerberg Initiative. So thank you very much, and we will get the show on the road. So it's my pleasure to introduce Alex and Sarah. So Sarah is going first, and then Alex. They're going to tell us all we need to know about ACA as we kick off this this historic symposium. So thank you very much.
Thank you, Gordon, for that lovely introduction, and it's a real pleasure to be here. And it's just fantastic that we have this meeting organized, even if it's virtual for the time being. So I'll spend the next few minutes talking a little bit about the HumanCellAtlas project and the history and how we've organized ourselves so far. And then I'll hand over to Alex to finish off, and then we'll have questions at the end. So the aim of the project, as I'm sure you all know, is to create a comprehensive reference map of our cells, basically the cells in the human body. And of course, this is important because cells are the fundamental unit of life, and we need to understand them. And understanding them will also have implications for diagnosing, monitoring, treating health and disease. And you can think of this sort of map of the cells of our body also as a periodic table where, in terms of the components that make up tissues. And those can include cells from many lineages, basically vascular, stromal, epithelial, myocytes, et cetera, and together, basically, these components contribute to the tissues, and you can see that we've organized ourselves in the community according to different tissues and organs that we have particular interests in, and that's shown at the bottom of this periodic table in terms of the bionetworks. Here you see different organs and systems: adipose, liver, lung, musculoskeletal development, et cetera, also organoids. And then we also have working groups in terms of both experimental standards and technologies, for instance, and computational in terms of the analysis working group. And so those groups have really evolved over time. And Gordon also mentioned other working groups. We've had ethics from the beginning, equity from the beginning, and so on. And you know, we invite people who are interested in those working groups to join, and also evolve other working groups and bionetworks and so on. And historically, basically, we had our first big kind of consortium kickoff meeting over 5 years ago now, in October 2016. And since then, the community has grown and it's evolved. And in particular, now, over the more recent past, we've also had regional meetings. The Asia meeting has had several installations, and it's really great that we now also have a Latin America meeting that I don't know if you can see my cursor, but I'm pointing to September last year here. And of course, now we have the African meeting here. and we had one meeting in Africa before on equity in Addis Ababa, but it's really great to have this meeting virtually now. And the benefit is, of course, that people don't need to travel and everyone has easier access to participate. And I want to emphasize, as I've said before, that we're a growing community and we're an open community. We're now over 2,000 members in 80 countries around the world. Sorry. 1,200 research institutes. And here at the bottom, I want to point out this URL here, www.humancellatlas.org/join-HCA. And there's a very easy signing up process with a simple form. So if you're interested in this project, and then please, I warmly invite you to take a look there and sign up, and you'll get the newsletters, information about the meetings, updates, funding updates, and so on and so forth. As I've mentioned before, we feel we are a completely scientist organized grassroots community. And we're now really truly global, and the global network has these geographical communities, and sort of going back in time, as I've mentioned, the HCA Asia regional network was established a few years ago and has had several annual meetings, and that now has over 400 members. Then the Latin American community has grown. You can see almost 100 members. And we're really delighted to now have the HCA Africa Regional Network that you're building here. And thanks to Gordon and others. And this, there are 28 members now, and I hope that many of you will sign up now that you've seen the URL, and it's very easy. So I've mentioned before that we have 18 biological networks, and I'm sure there will be many more in the future. And so far these have crystallized around coordinators and groups who are interested in these particular topics. And they include many of the major organs, also adipose, breast development, and so on. You can see here. The coordinators are listed here, and then there are many groups that are members. And so, in case you're interested in understanding the cells and their molecular fingerprint of any of these systems, please do take a look under, and there's more details here, humancellAtlas.org/areas of impact. And I'll now hand over to my partner in crime, Alex Shalek, who will take over from the next slide. And Alex, you can control them. Yeah.
Thanks, Sarah, and thanks everybody for having us this morning or this afternoon, depending upon where you are in the world, or maybe even this evening. It's awesome to be here. I really wish I was there in person. Hopefully we'll have the opportunity to do it soon. But if nothing else, maybe this meeting will give us an opportunity to get to know one another, where we already have informed bonds, and to start to plan how we would do something in person in the not too distant future. So as Sarah brought up, this is a relatively nascent grassroots effort that involves people from around the world. There have been a number of different communities that have been organizing the generation of atlases, which really mean, sort of maps of the single cells that come from various tissues. As Sarah pointed out, there are multiple different organs that are currently being profiled, and many different samples that have been looked at, comprising over about 53 million cells at this point. Yes, I'd like to be able to click and proceed. And so, there's a more specific breakdown that we can provide here, but one of the things that I think is important about highlighting this is to point out that, these are far from complete. So there are, early data sets that exist telling us about the multiple different parts that exist within each of these individual tissues. But I would also highlight that these are very large data sets that anybody can gain access to. And many of the things that the HCA has actually done, and the community has done, is to put these things in anonymized formats up on public portals, so that if you're particularly interested in the heart or, for example, the gut, you can go in and look at the data sets that are related to that and start to look at the properties of the individual cells that comprise those tissues. So there's the clicker. And I'll point out that one of the big things that we tried to do in the context of the recent pandemic, which is keeping us from being together in person, has delayed this meeting, is to begin to use this information to help us understand the cellular and molecular base of COVID infection. So in many of these different tissues, there are large references that will help you understand, what COVID might be doing in severe disease, mild disease, and different target tissues. And so I'd refer you to look at those. I think the biggest thing that I wanted to point out this morning, and sort of giving you an overview of the structure of the HCA before we dive into some of the science, is about some of the networks and groups that exist that help support this. I want to point out that the tools and methods that are powering the HCA are really these incredibly powerful molecular approaches that let you look at the properties of individual cells at unprecedented resolution. So a lot of it is the ability to sequence all of the RNA that exists in cells to look at the epigenetic state, to measure multiple different proteins at once. And these technologies can be applied to the study of human health, but also to human disease, and also to a number of other questions that might be of relevance to your own research. For example, it's been used by a number of individuals to look at the properties of malaria parasites. So one of the groups that we wanted to highlight is the Standards and Technology Working Group, and Holger will be here later today leading a session. They're sort of in charge of helping to establish the relevance of different methods, establishing best practices, performing benchmarkings, providing QCs, and they're incredibly useful and helpful. Their emails are listed on the HCA website. And if you're thinking about getting started and trying to figure out where to begin, they're a group of individuals which you can email or contact. You can also just directly contact me and I'll put you in touch with anybody or try and answer your questions. But there is a group that's really focused on the methodology and how well it works. And so I'll point out once again, these are broadly applicable, and I would encourage you to engage with them so that you can begin to think about how you would apply these tools to your research if you haven't already. The second thing that I point out is that the HCA generates a tremendous amount of data. If you think about it, fundamentally, we are usually sequencing individual cells. And so in the past, many studies have sequenced individuals, and one individual has been one sequencing run, so one set of RNA expression. Here we have the same information for every cell, so we get these enormous data sets that might be millions of cells across all of the genes that they express. So imagine something that's a million in one dimension by 20,000 the other. And that means that there becomes some incredible computational challenges to really figure out how to analyze and wrangle this data so that you can understand what it's teaching you about the underlying biology. And so there's also an analysis working group led by Donna Pierre and John Marioni, who organize meetings and jamborees to help people think about existing challenges, compare best methods, and really try and figure out how to extract as much information as possible from this data and overcome some of the technical challenges associated with measuring from very small inputs. And so I would similarly encourage you, if you're interested in computational work, to engage with Donna or John, or to email me. One of the great things about the HCA is because all this data already exists and is out there, it's an opportunity for you to engage and start working with it now, and even use it to reanalyze some of your own data sets. And I think you'll see as we go through the talks some examples of how some of the existing single cell data can already be useful to begin to generate hypotheses on existing data sets. The group I personally have been very involved in, along with Musa and Partha, is the Equity Working Group, and that's the guys through which we've worked with Gordon, with others, as well as in some science. I think that the HCA, because it's grassroots, has always envisioned this idea that we would engage people from all around the world, and that we would fundamentally try and create a reference that was useful to all of humanity. This is something that wasn't an afterthought. It actually showed up in the initial meeting in London back in 2016. And even in the white paper, we have a section dedicated specifically to equity. Within the Equity Working Group, we've really been trying to figure out how to engage scientists around the world to make sure that the HCA is not only representative in terms of the individuals that it profiles, but that also it has representative participation. That we're not doing helicopter science, that this really is driven by local scientists tackling problems of local relevance. And another thing that we've really paid a lot of attention to is data benefit, trying to make sure that the data that we're generating benefits the communities that are generating it, and that it doesn't accrue, to specific communities. And this is a place where, we're really interested in engaging and learning from each of you, so we can figure out how to help make sure that that is in fact the case. As was pointed out by Gordon, we've done a few meetings towards bringing this together. We've gone to many different places to do roadshows and trainings, teaching people about experimental and computational methods. We're looking forward to doing one, and then several after on the continent. But if you're interested in some of these things and how we might engage, please reach out to me, to Musa, or to Partha. We really do believe in equity in action. And by that, I mean getting boots on the ground, engaging with scientists, doing cross-training, really trying to support individuals and starting to use these technologies for their own research and becoming active members of this community. And finally, as you might imagine, trying to organize something across the entirety of the globe brings about some incredibly complicated ethical issues. And so you'll hear from some of the individuals that are involved in the ethics working groups that are trying to think about some of the ethical considerations that exist when you're trying to enable collaborations across multiple different individuals across multiple different continents. And in each instance, as you'll hear from Partha when he gives this talk later, we're really focused on being careful and being thoughtful about these things. We don't have all the answers, but we're interested in learning and in listening and trying to make sure that this project is really uplifting all of humanity as opposed to exploiting any individual. So lots of people that are present to try and help deal with some of the government's documents, to try and work with local funders or local boards to help make sure that data can be shared, but is done so in a way that actually supports each community that's helping to generate it. And so I'll highlight that we'll be talking about the HCA, which is sort of this healthy reference. This ties in very nicely with parallel efforts that are happening in the context of disease. We recognize that it's really important to understand what is healthy, to understand what actually is going wrong when you have a specific disease, because it helps you understand what are the molecular and cellular features that you might want to target, or biomarkers that might help you monitor progress. And so, while there is a big emphasis here on trying to figure out what baseline is, all these same technologies are equally applicable to studying disease. And in many instances, work on disease has actually happened before some of the healthy references. And we think that they're valuable and go hand in hand because it's hard to understand what's changing without that baseline. But while we'll talk primarily about healthy, all of us are deeply invested in not only understanding health, but also disease. And so I'll just conclude there and thank you all for joining us. Delighted to be here, wish I was there in person, and excited to talk about how we might partner to continue to do science and to build new collaborations and move things forward. And if you ever want to reach me and talk about HCA or talk about some of the methods or who to connect to, it's just my last name at mit.edu. And looking forward to the rest of the symposium. Thanks so much, Gordon, for having us.
All right. Thank you very much.